Hi, it is my pleasure to present our work on an intrinsically interpretable neural network architecture for sequence-to-function learning. So, sequence-to-function models attempt to predict the functional output of specific DNA sequence. And usually we will do this with a neural network. And these models have gotten incredibly powerful in recent years. So here's a figure from the recent Informer paper where the experiment is in green and the Informer predictions are in blue. And it is almost a perfect match. But I mean, what are we actually hoping to get from these models? So this figure just basically shows us that we can reproduce data that we already have. Well, one thing we can hope to get is the ability to predict functional readouts from arbitrary input. So this is very important when we're trying to do variant interpretation or designing synthetic sequences for some new functions. But another thing that we might hope to get is that if we have a very powerful model that understands biology, that it can explain it to us. So instead of producing variant interpretation kind of output, it can actually produce output that is of this form on the right, which is kind of an explanation for how cells acquire their self-identity in terms of the transcriptional regulation that is required. So one interesting example of really using the powerful neural network sequence to function learners in order to do exactly that is comes in from this paper by Sarah Mustafavi et al. And called Deep Learning and Immune Cell Differentiation. And in this paper, the authors propose AI-TAC, which is a CNN model and an interpretation framework. And so the architecture is actually fairly simple. It's just 3 convolutional layers and 2 fully connected layers, but we know that was extensively optimized, so there was apparently no gain to be had for making it more complex. The prediction task itself is chromatin accessibility across cell types, and the objective, interestingly, is correlation, because we're really, both the authors are really interested in getting cell type specificity right. But the scientific objective is to understand immune cell differentiation. So how do you go from a prediction model to biological insights? So AI-TAC proposes to do it in several steps. So you can map the initial convolution kernels to TF motifs. You can track the final vector activations down the TFs using a propagation framework like DeepLift, which is what they used. And when you put all of this together, you can get this motif to cell type map where we have the motifs here on the rows and the different cell types in the columns, and the heat map indicates, and its assigned value, it indicates the contribution of this motif to this kind of cell, to the identity of that cell. And it contains kind of a lot of things that we might expect. So, for example, looking at this module that is highly associated with B cells, it contains well-known regulators of B cell lineage commitment like PAX5 and EDF1. And for example, here's the same thing with ROR gamma being important for ILCs. Now, so what does this really say about the overall interpretability of these neural networks sequence to function models? So we can see the first layer kernels are actually interpretable, and we can actually compute the relationship between the first layer kernels and the final output. Okay, but this leaves us all of the internal layers. What do they do? So we can the answer to that can be kind of a fun answer, that they compute interactions between TFs and motifs and really compute like the complicated intricacies of regulation and motif grammar. And there's also some boring answers. For example, maybe all they do is refine motif or just do some kind of calibration from scores to binding probabilities. And the thing is, even if we can agree that in some cases we are looking at TF interactions, it is actually quite difficult to look at your neural network and just look at the parameters and even understand what kind of interaction, nonlinear interaction it computes. For example, we could be looking at a synergistic interaction, kind of an AND function, or saturating interaction is more like an OR function. These are both different from linear, so you need a nonlinear model to capture these, but it's not often obvious which of these is being computed. So for our work, we actually use the exact same data set as the AI tag paper. So I'll go a little bit into more detail regarding the data. So this is a developmental ataxic data set. There's 81 immune cell types, and they're actually arranged in this kind of developmental tree. One thing that's, even though there's, of course, a lot of ataxic data that you could use, one thing that's really great about this data set is that in the immunology space, a lot of the key developmental transitions have been studied for a long time and are really well characterized in terms of like the transcription factors involved and the direction of their effects. So the data consists of 7 main lineages. And the cross-lineage prediction is much, much easier than kind of predicting the saddle variation between cell types within a lineage. And this was actually already pointed out in the original AI tag paper, where you can see sort of the real OCR activity has some variation within lineages that now, for example, looking here, that now becomes quite washed out in the prediction space. So most of our experiments are going to be done in this 7 lineage setting. And we'll come back to the full data set at the end. So one kind of really simple thing you can ask is, we have a powerful nonlinear model. How much better does it do than just like a simple linear model? And for a simple linear model, we can take the elastic net regression and just max motif scores using all of CISBP PWMs. And so this depicts a performance gap. So this is done exactly the same way and for both methods, exactly the same cross-validation splits. So there's quite a bit of a performance gap. So one thing you might ask, so what's being depicted here is the R squared, so variance explained. You might ask, like, this seems a little low, especially for our neural network-based method. So the reason why that's low is that we're actually predicting the cell type Z score and not the absolute signal, and this is actually just a much harder problem. And okay, so a linear model definitely is worse, and as expected, but it's important to keep in mind it has some advantages. So it's convex, and in particular, that takes kind of all of the training dynamics out of the interpretation, right? You always get the same result. It's actually really easy to select hyperparameters. And the output is immediately interpretable. Like you get the cell type to TF map essentially for free because those are the coefficients of your model. So we want to design a model that really fills this performance interpretability gap. So we want to increase the capacity of our model and improve performance. But we want to do it in such a way that maintains basically the interpretability that we see, the immediate kind of intrinsic interpretability we see in the linear model. So the main idea is that we're not going to have any uninterpretable hidden layers. So every layer actually references TF activation. And all we're allowed to do is modify it. And the layer construction is biologically inspired. And so we call our model totally interpretable sequence to function model. Here's the model, and I'll take you through it step by step. So one of the inputs to the model is a set of convolution layer kernels. So we use CISBP for this. So these parameters are initially frozen, but they can be optionally unfrozen later. The other input is the DNA sequence. Of course, it's one hot encoded and amended in a couple of ways. So we use a positional embedding to account for the fact that motifs in the middle of attack signal are more important than towards the ends, but the decay is actually quite data set dependent. And we also use an attention module that allows some flexibility in motif activation without losing the motif identity. So these two inputs are passed through standard convolution. In the second step, we have a per channel trainable sigmoid activation. So it introduces nonlinearity, but it's per channel. So the channels are TFs and they're kept that way. Basically, the motivation for this is that common convolutions are not actually interpretable as motif binding; they're just scores. So we output the output of the convolution through sigmoid activation, and it has two trainable parameters, a scale and bias, and now everything is kind of like on a 0 to 1 scale. After that, the next layer is a trainable pooling, and basically this is a pooling method which interpolates between max pooling and average pooling. And the biological insight for this is that, well, generally we don't actually know whether or not we should do max pooling or average pooling, and which one would be best. You could try to determine this empirically, and this way we can learn it from scratch. But the idea is that if we have a transcription factor has low specificity, we can have all of these motif matches, but it's unlikely that it's actually, occupying any one of them at any given time. And so max pooling actually would give you false positives, and average pooling might be better. But for motifs that have a really high specificity and occupy their binding sites with high probability, max pooling is actually sufficient. Okay, after the max pooling, so the length of the sequence now has been reduced. So we now just have our channels, which is the TFs, and have been pooled. And we have one more layer, and it's we call it's the interaction layer. And basically what the interaction layer does is it produces a mask for each TF using the other TFs. So basically the contribution of each TF is reweighted based on what other TFs are present. Okay, and then in the final layer, the output is computed as a linear function of motif activations. And importantly about this model, the number of channels is always exactly the same. It's always equal to the number of input motifs. Motif identity is preserved. So the incoming channels all had names, and in the final layer, the names are still referencing the same TFs. And therefore, the final layer coefficients are immediately interpretable as a TF to cell type map without any post-processing. So we can look at the performance of this model. So looking at the same data set predicting cell type z-scores, what we now look at is the change in R squared, so not the absolute, but the change in R squared over AI attack CNN, which is a baseline. And we use that as a baseline because it was actually optimized on this data set for this task. So the architecture was we retrained it from scratch, of course. And so what we can see is that the full model does actually outperform the baseline CNN. And each layer, doing these ablation tests, each layer does actually help improve performance. Importantly, the biggest difference being made by the interaction layer. So biggest gap here, adding the interaction layer, going from yellow to green. If we allow the motifs to be fine-tuned, we could actually outperform AI attack in all of the different configurations for this model. And in this setting, we actually find that this attention module we included to allow for some flexibility actually no longer helps. One thing that you might worry about is that when you fine-tune motifs, they lose their identity, right? And this would, of course, impede interpretability of the final layer. We find that that is not the case, and actually 92% of motifs map back to themselves, and the rest actually map to another motif in the same family. Okay, so we said the final layer is immediately interpretable, and this is actually what it looks like. So we have very much like in the AI attack figure, we have cell types and we have motifs here. We're just showing the top 5 TFs for each cell type, and the heat map we get is in fact very, very similar to what we get with AI attack. So for example, we get EBF1 PAX5. We don't get like duplicated filters for the same TF. It's just what we put in. We get war gamma for innate lymphocytes. So all of the sanity checks are there, and we can also look at the biological interpretation of some of the intermediate layers. So we have this trainable pooling, and we had a very specific hypothesis about what it would do. And the hypothesis was that the pooling parameters actually really should be correlated with information content. If you have high information content, it should prefer max pooling, low information content, average pooling. This is actually what we get. So this is the pooling parameter phi versus information content. They're highly correlated, though it's not perfect. And just here we've highlighted some of the important transcription factors for predicting B cell identity. So we can also interpret the interaction layer. So here's a plot depicting kind of the overall interaction strength with the overall final layer coefficients for the TFs. And one thing we notice is that, in fact, they're different. We have a lot of these interacting transcription factors, which influence how the other transcription factors are counted, but that themselves don't predict anything about cell type identity. And actually, one of the top things is in this category is CTCF, and I think this is very intuitive. We know CTCF is very important for interpreting, kind of for the biochemistry of interpreting DNA sequence. But of course, being a ubiquitous protein, it is not, highly predictive of cell type identity. Zooming into the actual interaction matrix, we're just plotting the interaction coefficients between some of these top interactors and all of the other TFs. And one of the things so this is signed with blue being negative and red being positive. And one of the things we will note is that this heat map is really kind of quite dominated by blue. And blue is actually a negative interaction. And a negative interaction translated in a biological sense is actually just a saturation effect. So basically the simple, and this is very striking for CTCF, has a lot of interactions and they're all negative. And basically the interpretation of this is that once I see a CTCF site, then nothing else matters about that region. Like that basically determines how it looks in my ataxic data. And in fact, if you go back and just do like a very simple marginal analysis, the presence of CTCF is the one thing that is most correlated with being a low variance site across cell types. It's just always open. Finally, the final question we ask is for our linear model we initially compared with, we used elastic net, which on top of all of the advantages that I listed, also has this nice advantage of being able to give you sparse models. So we asked, can we get actual true sparsity in the final layer of our neural network model? And we used an atom optimizer with the MCD proximal operator. This is actually a fairly recent development. And so you can tune up the sparsity, and in fact, you can get a slightly sparser model that is actually slightly more performant. You can look at kind of the coefficient dropout curve. So this is just on the innate lymphoid prediction, and so here's just over all the cell types. At the best prediction, at the lowest MSC, just the number of non-zero coefficients that we get. It is quite a bit smaller than the total number that we put in, and interestingly, you need kind of quite a bit more transcription factors to predict T cell states, which is something that people have noted before, that T cell regulation, there's not actually like a master regulator of the T cell state, so that's quite consistent with known biology. Finally, we want to go back to the full data set. And but instead of predicting kind of the individual cell types, we redefine a problem a little bit and we make a new prediction task. And it's predicting the difference between yourself and the parent. So it's actually, we're making a prediction along the edges of the developmental trajectory. And this problem turned out to be really hard. So anything we call a successful prediction, which is anything which has an R squared of about 0.05. And these are all the things that are colored on this tree. So for the most part, we only observe this between major lineage commitment points in this tree. And we have one exception, which is this CD4, CD8 split over here, which actually doesn't really induce dramatic changes in ataxic itself, but was relatively well predicted with our model. And going into the coefficients, we can ask, okay, well, what is contributing to this prediction? And actually, one of the top contributions is coming from this transcription factor, ZBTB7B, which is actually known as THPOC. And no surprise, it is actually on this classical textbook diagram of T cell differentiation. And not only do we get this correct as the master regulator of CD4 T cell commitment, so it's highlighted really for the CD4 branch, not the CD8 branch, the coefficient is actually negative, which also is correct. It is known that the way this transcription factor induces CD4 differentiation is by suppressing the CD8 program. And this is not a transcription factor that actually appeared in the lineage aggregate data set. So just finally, a few things that we tried that didn't work. Our model is completely agnostic to any grammar, right? So this is just, we're counting kind of like presence, absence, and none of the positional effects or how the transcription factors are positioned relative to one another. We did try to do this, but it didn't work. And so in what sense did it not work? So we had actually 3 metrics of success: performance, stability across folds, and kind of an external metric, which is recovery of known all cell type commitment TFs. And more complex models just tend to lose that final layer interpretability and stability across folds. And performance, of course, is actually okay. You can make very complex models that perform just fine. And just in conclusion, we're able to build a model can outperform standard CNN models, is intrinsically interpretable. And what have we learned? So modeling motif collaboration and saturation is critical for model performance. Motif database really contains sufficient motif representation, and it's enough to get a good model, though fine tuning does actually help. And in future work, we'd like to, really try a little harder on building position-aware models. And with this, I'd like to thank the funding, and I'd like to thank Sarah Mustafali, the author, AI Tag, for helpful discussions. And thank you for listening.